एवरीवन, तो आज हम पढ़ने वाले हैं आयरन का रोल क्या है इन लिविंग सिस्टम्स एंड हीमोग्लोबिन एंड मायोग्लोबिन का स्ट्रक्चरल फीचर्स एंड देयर एक्टिविटीज. सो द फर्स्ट टॉपिक इज द रोल ऑफ आयरन इन द लिविंग सिस्टम्स. सो द आयरन इज अ functional unit of hemoglobin and myoglobin and other metalloprotein present in the animal body. So let's see how it will present in different metalloproteins. So iron that is acting as a vital transition metal. तो आयरन एक बहुत ही इंपॉर्टेंट ट्रांजिशन मेटल है इन द लिविंग सिस्टम. इट इज़ वेरी इंपॉर्टेंट फॉर प्लांट्स एंड एनिमल बोथ. It is present in three well-characterized systems. First of all, in the heme proteins, non-heme iron sulfur protein and non-heme diiron proteins. So heme proteins means proteins that contains one or more porphyrin rings. This is a porphyrin rings with the iron. So the example of this metalloprotein is hemoglobin, myoglobin and cytochrome P450. And second is non-heme iron sulfur protein. Proteins which containing the iron sulfur complex. This is iron and the sulfur bridge and sulfur ligand. Basically assistants are present. In the examples are rubridoxin, ferridoxin, nitrogenase. And the third part is non-heme diiron kind of metal protein. So oxo-bridge compounds such as the carboxylates are present. This is iron connected by the oxo-bridge and other ligands are present. For example, hemerythrin ribonucleotide reductase and methane monooxygenase में रहता है जनरली. तो कुछ इंपॉर्टेंट नेचुरली ऑकरिंग आयरन प्रोटीन्स और उनका जो फंक्शन होता है लिविंग सिस्टम में वो इधर लिस्ट किया हुआ है. तो हीमोग्लोबिन, दिस इज फ्रॉम अ रेफरेंस बुक. हीमोग्लोबिन का मास होता है 64500 एंड इट कंटेन्स 4 आयरन एटम Fe इन द 2+ स्टेट एंड सोर्स इज एनिमल एंड दिस हेम यूनिट इज प्रेजेंट एंड इट्स मेन function is oxygen transport. Myoglobin containing 1 iron atom Fe in 2 state animal's body it is found and it containing heme and its main function is oxygen storage. In cytochromes 4 iron atoms are present and Fe in the 2 state and it is found in plants, animal and bacteria and hemes are present and its main function is electron transfer. Ferridoxin contains 2 to 8 unit of iron and Fe present in Fe2 and Fe3 state and it is present in bacteria, plants and animal and here non-heme kind of protein this is and its main function is electron transfer. Rubridoxin containing 1 iron and Fe in the 3 state it is found in bacteria. This is a non-heme protein and its main function is electron transfer. In the ferritin 20% of iron is present and Fe in the 2 state and it is found in animal body and non-heme protein. It will store the iron. Transferrin, it contains 2 iron atom and Fe in 3 state it is found in animals and non-heme protein and this will scavenge the iron. Hemerythrin, it contains 2 iron Fe in the 2 state it is found in marine invertebrates and non-heme protein and its main function is oxygen transport. Iron molybdenum protein containing 24 to 36 unit of iron and it is present in nitrogenase enzyme and this is a non-heme protein and its main function is nitrogen fixation in the bacteria. Catalase, it contains Fe3 plus and it is found in living organism. This is heme kind of protein and decomposition of H2O2 is its function. Peroxidase containing Fe3 and it is found in living organism. It is a heme protein and its main function is decomposition of H2O2. Now we will discuss about the chemistry of the heme proteins. So हम डिस्कस करने वाले स्ट्रक्चर, फंक्शन और फिजियोलॉजी ऑफ हीमोग्लोबिन एंड मायोग्लोबिन के बारे में. So let's see. This is a overview what we will learn in the next portion. So this is a molecular architect of respiration. This is hemoglobin and myoglobin. So they are structurally distinct protein but specialized physiological roles in oxygen transport and storage. First of all, we'll learn about the hemoglobin. So this is acts as a dynamic transporter. Mainly this transport the oxygen. And this is a tetramer. This is a heme, this is heme, this is heme, this is heme. And it is surrounded by the globin protein. Okay. So this is a tetramer containing alpha to beta 2 unit. This alpha to beta 2 are protein chain, globin protein chain basically. And this is a kind of a tetrahedral geometry. Means if we say this kind of tetrahedral, they are present in tetrahedral arrangement. If we connect this 4, they are present in a tetrahedral arrangement. So this is a heme B unit in the active site. So iron 2 coordinated to the protoporphyrin 9. Already it is discussed in the previous lecture. Or tetrapyral ring derived from the porphyrin. And in the myoglobin, the stable storage unit, its main function is to store the oxygen. It is a monomer. It is containing only one iron and porphyrin plus this globin chain. So this globin pocket have very important role. So ये जो हेम है, इसको इनकैप्सुलेटेड करके हाइड्रोफोबिक पॉकेट तैयार करती है, ये जो ग्लोबिन चेन, and it will prevent Fe2 to Fe3 oxidation. So basically, if this is not, the same unit not surrounded by this globin chain, it will bind to oxygen and it will convert to hematine. And this hematine iron is present in 3 state. After that, we will discuss about the T form and the R form of hemoglobin. So this is 10 state and this is a relaxed state. So deoxy state, which is also called a 10 state, because iron in the +2 state and high spin complex. So iron is out of this cavity of porphyrin ring due to larger size. So that's why this is in the 10 state, and the Fe bond length about 218 picometer, and geometry kind of a square perimeter. And after the oxygenation, which will trigger it to convert to the relaxed state, which is oxyhemoglobin, where this iron is low spin and size will decreases, so it will feed the cavity of porphyrin properly. And bond length of Fe nitrogen will decreases, and this is a octahedral geometry, and here the oxygen will attach with it by band arrangement. So massive conformational change facilitated by 70 picometer of shift. So this distance is 70 picometer. So it is apart from the square plane is 70 picometer. Okay. So when it is converting to this, so iron approximately shifting 70 picometer towards the ring. So which will help to massive conformational change, and that will rapture the salt bridge interaction present in 10 state. उसके बाद हम डिस्कस करने वाला है कि बाइंडिंग डायनामिक्स एंड कोऑपरेटिविटी ऑफ हीमोग्लोबिन एंड मायोग्लोबिन. सो इफ वी प्लॉट ऑक्सीजन सैचुरेशन वर्सेस ऑक्सीजन प्रेशर, if we plot that, we'll get for hemoglobin a sigmoidal plot and monotopic plot for the myoglobin. So basically this sigmoidal plot is obtained due to the cooperativity effect. We'll discuss about it later stage. So basically at the low oxygen pressure, the hemoglobin will lesser binding tendency with the oxygen, otherwise myoglobin has higher binding tendency with the oxygen. But at the high oxygen pressure in the lungs, the hemoglobin will bind strongly with the oxygen in comparison to the myoglobin. And the cooperativity effect means that suppose there is a 4 unit of this iron and porphyrin globin chain unit in the hemoglobin. So if it is bind first, then it will help other unit to bind faster. So this is a homotopic allosteric effect. So binding of one oxygen will increase the affinity of remaining 3 subunit to bind faster. And another is that Bohr effect and chemical protection. So Bohr effect is about related to the pH. So this is in the basic pH, this is in the acidic pH of hemoglobin. So in our cell tissue, there is a low pH, so high carbon dioxide concentration and acidic concentration. So that stabilizes the 10 state and triggers the oxygen to release from the hemoglobin. And the distal histidine, this is the distal histidine unit. It has very important role. So it forces CO, that carbon monoxide, which acts as a poison for our body, which this group will force the CO group to bind in a bent way. Okay, in a bent way. We know that here oxygen will bind, but if in our body, if CO enter, it will bind here. So if CO is entering in this in a perpendicular way, means in a 180 degree, 90 degree angle, then it is very dangerous, because this is a strong pi acceptor ligand and it will poison our blood and it can cause the life of humans. So this distal histidine will protect the hemoglobin from that, and it will bind bend shape and it can be removed in other ways. Carbonate buffering system. The carbon dioxide acts as a buffer in presence of carbonic anhydrase, is converting to the carbonic acid, which will maintain the blood pH. हम पहले ही डिस्कस कर चुके हैं. There are 2 metalloproteins which are very important. One is hemoglobin, another is myoglobin. So hemoglobin acts as a transporter. So it is a tetramer unit. It is containing 2 alpha unit, 2 beta unit, and its molecular weight about 64500 Dalton. And its function is picks up oxygen from lungs and delivers to the tissue. And the myoglobin, this acts as a storage unit. This is a monomer. This is single polypeptide chain and containing hem. The molecular weight about 17800 Dalton. Its function is it will store oxygen tissue for the metabolic action. So we can say this is acts as a battery of cell. So in the last lecture we already discussed about this, about the porphin to hem B. This is a porphin ring. All these pyrrole ring are connected by the methylidine linkage. So this is forming a porphin ring. So this is a basic skeleton for the hemoglobin or myoglobin. After that it will convert to the protoporphyrin 9 when we substitute the different ends with the methyl or alkene or the carboxylate group. And in the hem B it will release 2 protons and the iron is bind to it, and this is finally forming the hem B. सो हमारे दिमाग में एक क्वेश्चन तो आता है कि ग्लोबिन चेन इतना इम्पोर्टेंट क्यों है. अगर सिर्फ एफ होता, पोरफाइन रिंग होता तो वो भी चल जाता, बट ग्लोबिन चेन व्हाई इट इज़ प्लेइंग एन इम्पोर्टेंट रोल. सो लेट्स सी. सो ये जो ग्लोबिन चेन है ये स्कैफोल्ड की तरह काम करता है. दिस इज गिविंग द स्ट्रक्चरल स्टेबिलिटी टू द दिस आयरन पोरफाइन रिंग, बिकॉज all this globin that is attached to the proximal histidine, and another histidine is that here this is F7 unit that is called a distal histidine, and here another imidazole ring will present, and that system also that containing the another globin chain, and this globin chain in the deoxy form, here CO minus state and this in the NH3 plus state. So this will generally forming the Salvage interaction, and similarly in this way here this is CO minus, this is NH3 plus. So this will also form the Salvage interaction. So this overall stabilize the system of hemoglobin and myoglobin unit. So in the quaternary structure the Salvage interactions are present. I already discussed about it. So hemoglobin में tetramer of hem unit रहता है. This is 1, 2, 3, 4. So each one will do total 2 interaction. Means each hem unit total 2 salt bridge. Okay. This is 2 alpha unit, 2 beta units are present, and each hem unit will give 2 Salvage interactions. So total interaction will be 8 distinct Salvage interactions are present here, and which will stabilize the whole tetrameric structure. So ये जो हेम है इसके अराउंड जो ग्लोबिन चेन है ये एक हाइड्रोफोबिक पॉकेट की तरह काम करता है. तो एक्चुअली वाटर और एनी H2 और any polar solvent can't enter inside this cavity, so this will protect from the polar molecules, this globin chain. And another molecule is the 2,3-DPG, which is 2,3-diphosphoglycerate. अगर एग्जाम में आए तो इसका रोल ही पूछते हैं, बट कभी भी इसका क्वेश्चन नहीं आया. बट फॉर नॉलेज पर्पस, सो इसका जो रोल होता है वो पॉजिटिव बीटाजेन का जो पॉजिटिव रेसिड्यू है, मतलब आपका पॉजिटिव रेसिड्यू NH3 प्लस, दैट विल बाइंड टू इट, लॉकिंग द स्ट्रक्चर इन द डीऑक्सीजनेटेड स्टेट. इसके साथ बाइंड होके वो ये स्ट्रक्चरल स्टेबिलिटी देने में हेल्प करता है. अब हम डिस्कस करने वाले हैं कि कैसे CO पॉइजनिंग को इनहिबिट करता है ये जो डिस्टल हिस्टिडीन यूनिट. आई ऑलरेडी डिस्कस दैट व्हेन CO इज बाइंडिंग, दिस इज अ पावरफुल pi acid ligand, and this will bind in a 90-degree angle with the iron, and its affinity about 25,000 stronger than the oxygen binding affinity. So the distal histidine plays here an important role. If the distal histidine not present, then our body cannot able to live in presence of CO. So this distal histidine will force the CO to bind in a bend fashion with the help of the steric hindrance, and that bend fashion binding is not that much strong, so it will more favorable to bind with the oxygen in the bend fashion. So I already said that CO is a powerful pi acid ligand. The distal histidine forces sterically unfavorable bend configuration allowing the body to resist the endogenous CO poisoning. So that's how the distal histidine will prohibit or forces not to do the CO poisoning in our body. अब हम डिस्कस करने वाले हैं टी फॉर्म और आर फॉर्म के बारे में इन द हीमोग्लोबिन. सो इन द डीऑक्सी हीमोग्लोबिन it is present in a T form or TN state. So basically this is a porphyrin ring plane and iron is above 70 picometer above the plane and iron and nitrogen distance about 218 picometer. So iron present in a high spin state T2G4 Eg2 and its size is larger, so it can't able to fit in the cavity. And in the oxyhemoglobin when oxygen binded to the iron, तब क्या होता है, तब आयरन कन्वर्ट हो जाता है लो स्पिन कॉम्प्लेक्स में iron 2 in the T2G6 state. So its radius will decreases, so it will fit to the cavity of porphyrin ring. और ये जो 70 पिकोमीटर का मूवमेंट होता है आयरन की नीचे की तरफ, that pulls by the 8 histidine that will rupture the salt bridge interaction. Means here the salt bridge interaction is present, that will ruptured by this pull effect. So in the oxy form the salt bridge interaction is absent, but in deoxy form to give the structural stability the salt bridge interactions is present. So that is why this is called a relaxed state or R form. Now अगर ऑक्सीजन का बाइंडिंग और मॉलिकुलर ऑर्बिटल के बारे में बात करें तो ये पोर्फायरिन रिंग का प्लेन है, आयरन है, और ये जो बाइंडिंग होता है ऑक्सीजन का इट इज़ अबाउट 136 डिग्री एंगल. और IR स्पेक्ट्रोस्कोपी के बारे में बात करें तो नॉर्मल ऑक्सीजन स्ट्रेचिंग फ्रीक्वेंसी अबाउट 1550 सेंटीमीटर इनवर्स, बट the oxygen present in the molecule is in the O2 minus. This is a superoxide, okay, and iron is the 3 state. So first of all it will bind in the Fe2 state with neutral oxygen. After that it will convert to Fe3 state and O2 minus, means charge transfer takes place from the iron to the oxygen. और इसका जो स्ट्रेचिंग फ्रीक्वेंसी होता है वो अबाउट 1106 सेंटीमीटर इनवर्स. और मैग्नेटिक प्रॉपर्टी के बारे में अगर बात करें तो हीमोग्लोबिन वो डायमैग्नेटिक होता है बिकॉज T2G6 कॉन्फ़िगरेशन, दिस इज फील्ड कॉन्फ़िगरेशन, बट इन द डीऑक्सी फॉर्म दिस विल गिव द पैरामैग्नेटिक सिस्टम. सो द सिग्निफिकेंट चार्ज ट्रांसफर इज ऑब्जर्व फ्रॉम द आयरन टू ऑक्सीजन. अब हम डिस्कस करने वाले हैं कि जो दो हिस्टीडीन मॉलिक्यूल है उनका क्या रोल होता है. तो ऑलरेडी वी डिस्कस अबाउट एवरीथिंग. तो ये जो E7 है ये गेट कीपर की तरह काम करता है. So it will stabilize the oxygen by forming the hydrogen bonding with it and also prevent of the CO poisoning. Okay, so it has very important role. On the other hand, the proximal histidine that will act as an anchor, and this is coordinated to the metal, transmit the mechanism pull effect, so which will help in the structural change. Structural change में help करता है. So it will easily converting to the deoxy to oxy form. नाउ ऑक्सीजन बाइंडिंग का जो जियोमेट्री है उसके बारे में डिस्कस करते हैं. तो आई ऑलरेडी सेड दैट इट इज़ अबाउट 136 डिग्री एंगल बिटवीन FeO बॉन्ड एंड द लेंथ इज अबाउट 197 पिकोमीटर, बट इन द डीऑक्सी स्टेट द डिस्टेंस इज 282 पिकोमीटर. और ये जो डिस्टल हिस्टीडीन इसके साथ हाइड्रोजन बॉन्डिंग करता है वो इस स्ट्रक्चर को स्टेबिलिटी देता है. नाउ हम डिस्कस करने वाले हैं कि ऐसा भी तो क्वेश्चन आ सकता है कि अगर ग्लोबिन चेन नहीं होता तो क्या होता. तो लेट्स सी. सो द porphyrin iron in the 2 state when it is combined to the neutral oxygen, so it will just bind to it, no oxidation state. After that mu peroxo complex will form. So this is Fe2O2 and another Fe2 and porphyrin will come and bind to it and it will generate a peroxo linkage, both Fe in the 3+ state. This is also 3+ state. Now उसके बाद क्लीवेज होके फिराइल कॉम्प्लेक्स फॉर्मेशन करता है. तो ये जो ऑक्सीजन है इसका मिडिल से क्लीवेज हो जाता है एंड Fe इज कन्वर्टिंग टू द +4 स्टेट एंड बाइंडिंग टू इट इन अ दैट वे. दिस इज कॉल्ड अ फिराइल कॉम्प्लेक्स. उसके बाद जो डाइमराइजेशन होता है एंड हेमाटिन फॉर्मेशन करता है. ये जो फिराइल कॉम्प्लेक्स है उसके साथ Fe2 porphyrin अगेन बाइंड होता है और बोथ having the 3+ oxygen state and bonded by 1 oxygen. This complex is called hematin. Means अगर ग्लोबिन चेन अगर एब्सेंट है तो ये ऑक्सीजन के साथ बाइंड होके फाइनली हेमाटिन फॉर्मेशन करता है. तो ऑक्सीजन ट्रांसफर नहीं कर सकता ये हेमाटिन एंड दिस इज स्टेबिलाइज्ड इन द हेमाटिन स्टेट, सो इट विल इनहिबिट द बायोलॉजिकल फंक्शन. नाउ अभी हम डिस्कस करने वाले हैं कि कैसे नेचर ने हमारा बॉडी में डिफरेंट मेटलोप्रोटीन को तैयार किया ऐसे प्रॉपरली कि सारे प्रॉब्लम का एक सॉल्यूशन है. सो बायोलॉजिकल सॉल्यूशन है कि जो हाइड्रोफोबिक पॉकेट, मतलब ये जो ग्लोबिन चेन है सारे हाइड्रोफोबिक पॉकेट तैयार करता है, तो उसके वजह से लो डाइलेक्ट्रिक कांस्टेंट होता है, एंड दैट्स व्हाई द चार्ज सेपरेशन विल बी प्रिवेंटेड. एंड स्टेरिक इफेक्ट के बारे में बात करें तो ग्लोबिन चेन प्रिवेंट टू हेम सेंटर कम क्लोज टू ईच अदर. ये जो ग्लोबिन चेन है वो दो आयरन हेम यूनिट को क्लोज आने से रोकता है ड्यू टू द स्टेरिक हिंडेंस इफेक्ट. So by that I'll end this lecture. In the next lecture we'll learn about the other parts and other metal enzymes. Thanks for watching the video.